Greetings. I'm Elijah Jackson, a third-year biology student from Miami, Florida. Today, my presentation will be about the bacteria called Staphylococcus aureus, which is the unknown that I found during the DNA sequencing. To amplify a single copy of DNA, scientists use PCRs. PCRs include double-stranded DNA templates, nucleotides, primers, and DNA polymerase enzymes. PCR cycles begin by heating the DNA to denature the double-stranded template. After denaturation, primers bind to their target sequence on the single-stranded DNA. Primers are then extended by DNA polymerase to make the complementary second DNA strand. As the number of cycles increases, the number of DNA molecules increases exponentially. To determine the size of the DNA fragments, we prepare a gel with appropriate concentration of agarose fluorescent dye. We then pour the gel into an appropriate mold and allow it to set. Next, samples are loaded into a gel to begin current. Visualize the DNA fragments under UV lights. This process is used for the separation of DNA fragments, and it can also determine the exact size of fragments. The unknown that I found was Staphylococcus aureus. Staphylococcus aureus is a gram-positive bacteria, meaning it has a thick cell wall composed of peptidoglycan. It colonizes the nose, respiratory tract, and even on the skin. The sequence match and the naive Bayesian classifier both match because both are in taxonomic hierarchy. The only difference is the sequence match provides a SAB score, while the naive Bayesian provides a library. Also, Mercer screening weekly helps with empirical therapy. Mercer screening in many cases can be detected, but without it, many can go unnoticed. Also, its transmission is by direct contact between humans and mammals. It can also be transmitted by an infected person's cough or sneeze, releasing germs into the air, which we breathe in, and this is how Staphylococcus aureus colonizes in our respiratory tract. As previously stated, Staphylococcus aureus is a gram-positive bacteria. Also, its morphology is coccus. It causes a variety of diseases such as food poisoning, toxic shock syndrome, scalded skin syndrome, and many more. Everyone is susceptible to Staphylococcus aureus. It is also responsible for many infective and nosocomial infections. Epidemiology of Staphylococcus aureus includes gastrointestinal infection, and that's caused by episodes of food poisoning through enterotoxins, nosocomial infections. An example of this is MRSA. MRSA is a strain of Staphylococcus aureus and is commonly transmitted in healthcare environments. It can colonize by antibiotic exposure and surgical incisions. Another one, which is the most common one, is skin infections. It commonly colonizes the skin, but it can also cause infection if the skin is bruised or cut open. Pathogenicity of SRS. The pathogenicity of S. aureus. Abscesses are the manifestations of S. aureus, is where the site of infection begins. Polymorphonuclear leukocytes is the primary components of S. aureus that causes the disease. They can produce many antimicrobial agents that are skilled at killing bacteria but can also damage the host tissues, and then the formation of abscess begins. Methicillin-resistant S. aureus, also called MRSA, is an antibiotic-resistant strain found in S. aureus that causes infections. It is resistant to many antibiotics. Also, S. aureus antibiotic-resistant strains have reached epidemic amounts globally, especially in healthcare and community settings. The disease it can cause is minor skin infections such as abscesses, pimples, and carbuncles. This may lead to meningitis and also endocarditis.